You just say your name?
Miheer Sane.
Okay, great. Can you talk about your research, Miheer?
Sure. So we're studying one of the two regions of the brain, of the adult brain, that's capable of making new neurons. It was previously thought that there's no neurons produced after development. So the adult brain, once it's formed, that's it. But now these two regions have been found, and it provides hope for regenerative therapy and stem cell study. And so studying one of these two regions, one of the other studies found that the stem cells form what's called a pinwheel structure, and they talked about this as kind of a fundamental regenerative unit. Other studies had seen that with aging, there's a decrease in the number of neurons produced. So we wanted to see if this really is a regenerative unit, how does this change with aging? So we looked across the area within the brain which it's located, and we counted the number of these pinwheels, and we saw in a mouse model, from 3-month-old mice to 1-year-old, there was a dramatic decrease in the number of these pinwheel structures. The other thing we kind of looked at was that with these stem cells, they're very similar in terms of their contacts there's the fluid-filled ventricles of the brain, and they have a contact with that as well as with blood vessels, and that's very similar to the developmental stem cells. So we looked at the development of these pinwheel structures, and we were able to see, so in newborn mice, as they age, we saw decreased expression of the radial glial markers only remaining in these pinwheel structures. So kind of suggesting that these adult stem cells are very related to the developmental. And then the last thing is really just looking at the markers within the region. One of the problems is that there's no perfect marker for stem cells, but in order to have a therapy or anything, we need to have proper identification of these cells. So we looked through and we were able to see that there may be different populations with differential expression of these various markers. And I've really enjoyed doing this work, and through the private funding that I was able to get, was able to stay here over the summer and have the time really to do this research. During the year, it's very hard to fit everything in, but it was a great experience in the summer in terms of learning everything as a whole. I learned new imaging techniques to be able to take these images and really understand what I'm studying. And for the future, I'm interested in an MD PhD program. And throughout this year, I've been interviewing, and it's been a great experience to be able to share the work that I've done. I'm currently accepted to a few programs, and
Great.
My research and the funding that allowed it definitely helped.
Great,
make
fantastic.
that possible.
Excellent, thank you.